Hello, my name is Duane Johnson. This is July 11th, 2018. This is pouring agar plates. This is going to be a part of my aseptic technique and culturing video, so I just wanted to add this part in. I've already started 2 other agar plates that I'm going to be using for my, I think what's it called, the culturing of my cultures for when I go to put them in the agar plates. I'm going to go from quadrant to quadrant with the whole culturing and aseptic technique video and lab. So here's the first part. I'm going to grab my TSA, the soy agar, the triptych soy agar, I believe is what it's called. So lid's a little loose. I've already poured these first 2. I'm just going to pour half of this one because I think I need to save it for another one. This is the last one. So just like that, real quick. Half the tube. Should be good. Down here. And then I'm just gonna set that out, let it cool. It'll probably solidify, and then I'll pour that back or put that back into the refrigerator for later use. But for now, these are just the 3 plates that I'm going to be using for the rest of the aseptic technique and culturing lab video that I'm going to do. So next we'll take these inoculation loops. I'll put it into the nutrient broths that I have with my cultures, and then I'm going to start to spread them across the plates here. Each one of them are labeled E. coli, Saccharomyces cerevisiae, and Staphylococcus, can't exactly see, epidermidis, I believe is how it said. So thank you. Hi, my name is Duane. This is part 2 of the bacteria or the aseptic technique and culturing microbes. We're going to be doing the streak plate or plate streak or streak plating. We have our poured agar plates down here. We're going to take our inoculation loop out of the alcohol so that it's sterile. And wait about 20 seconds so that way we know it's sterile. In the meantime, just take my E. coli here. Go ahead and sterilize the tip of this. And this was the one that had the lip burnt through the sterilization process last time. Now, with my aseptic technique, I think I should remind myself not to just haphazardly place the lids onto the working surface. So, with that in mind, I'm going to try better to not do that. Alright, that should be sufficient. So now we have our nutrient broth, sterile inoculation loop. Oh, let's go ahead and take the, no, here, we'll do it like this. Go to the bottom. Sterilize the lip again. We're going to save this for later. So if you had noticed when I went to use the inoculation loop back into the agar plate for the streak plate, I went in a zigzag motion. Now I'm not sure if I had let the agar sit up long enough. We weren't really instructed or had any instruction given as to if the media needed it to be completely solid, still a liquid, or not, but learning experience for everybody, so we'll see, we'll determine if that was adequate or not. It should set up a little bit more, probably solidify a little bit more. I am, however, going to move my candle so that it's more out of the way of my forearm when I go to streak the plates. So my inoculation loop is back in the alcohol, another 20 seconds. We'll go from one other end of the quadrant that I had just gone into another quadrant into 4s, and hopefully we can isolate the bacteria a little bit better. So give it a little time, and then we're also going to do that for all of our other cultures as well. You know, I'd imagine that labs would have positive pressure rooms when they do all these types of experiments and stuff with other kind of pathogens and microbes and all that. So now we're going to go from the other quadrant. So we'll go from this edge. At least I can see where I'm at, though, in the media, so it's a little clumpy. This may not go as well as planned. Not sure if it's supposed to be broken up like that. This is my next culture, Staphylococcus epidermidis. think for future reference, it's worth noting how this inoculation loop should be used. Like inoculation loop, like horizontal over the plate, zigzagging, or like downward, vertically, zigzagging. Maybe I just didn't read that. Probably like this. This is the yeast, or Saccharomyces cerevisiae, nutrient broth. Pretty sure that's the way that's supposed to be done. That's it. Now they go to incubate for 48 hours, 24 to 48 hours. Take some pictures and upload them to the lab assistant.